Okay, so in class we didn't get to finish up the classification of bacteria. So this is going to be a fairly quick overview of the groups, the names of the groups, and what's kind of most important for you to focus on. So if you look up bacteria classification online too, you're going to see something slightly different. And also, the more you look, the more different sorts of things you're going to find. Bacteria are always, since like the 1880s when the gram stain was developed, gram positive, gram negative is what was used to separate the cells into two groups, and that is still used today, and it's still our top level. All right, so that's one thing you need to know. Just one thing we'll list up here. What sorts of things, in addition to what we have here already, what sorts of things do we use to classify bacteria? So obviously gram stain is going to be the top. And remember, gram stain refers to the cell wall, and make sure you review in detail the structure of the cell wall of the bacteria and what separates the two. So for gram positive, we started to do this already, so this is just a real quick breakdown. The gram positive bacteria, now another thing we could use, and you'll see this in some places, would be the shape of the cell, so like cocci or bacilli. That's not what we're using here. Okay, here we're using something else. We're using the concentration of guanine and cytosine to break them into two groups. So greater than 50% and less than 50%. So we call them the high GC group and the low GC group. So that's how we are breaking it down. Like I said, you'll see it other ways, but this is what we're doing. Another way of doing this too is going to be, in addition to shape, is going to be the oxygen use. So another different but common way to do this would be gram positive, cocci versus bacilli, and then for each of them, aerobic, anaerobic, aerobic, anaerobic, and kind of break them down. Those things are important, so how they use oxygen, those will be unique characteristics of different bacteria. But what we see is as we look at the genetics of the bacteria, those things are all over the place. Okay, so you'll have members of this group that are cocci and bacilli, members of this group that are cocci and bacilli, members of this group that use oxygen or don't use oxygen, and so forth. So those things are important in describing characteristics of bacteria, but they're not what we're using in our system of breaking it down, sort of more of the evolutionary relationship sense. So which is primarily genetic. So guanine cytosine is what's used. The Firmicutes group, sometimes also referred to as the bacilli group, although like I said, members could be cocci shaped. One unique thing about this group, or maybe two unique things about this group. One I have already written up here, is that outside the gram-positive cell wall, in some of them, is something called an S layer. Okay, so this is an extra layer of protein. So this S layer is an extra layer of protein that is outside the peptidoglycan. All right, outside the peptidoglycan layer, so if we had, say for example, our peptidoglycan layer, inside of that is the cell membrane. Okay, here's the cytoplasm of the cell. Remember, we have our teichoic acids, sticking up. 4 gram positive, the lipoteichoic acids also coming out of here. And now we're having, for some of these, we said, we're going to have this thing called an S layer. So it's actually a layer of protein, an extra protein layer, that's on the outside, the extracellular side. And we see that just in the Firmicutes. We don't really see that anyplace else. And not all the members of them, but quite a number of them do. So keep that in mind. Also, something that we're going to hit more in lab, specifically will be with the endospore production. Again, not all members produce endospores, but of the bacteria that do produce endospores, they belong into this group. So that's where the bacterial vegetative cell is under stress and is going to die. And then it packages up its DNA into this little very resistant spore, which will hang around for a very long time until the conditions are good, and then it can regrow its vegetative cell, which is details that we get into separately on the structure of and production of endospores. So Firmicutes are the low GC group of gram-positive bacteria, and a couple extra little things about them. Some have this extra protein S layer, some make endospores. Actinobacteria. These are the high GC group. That's the way we characterize them. Some, not all members of the group produce antibiotics, but it's noteworthy because there are some very important groups within this Actinobacteria that produce, like Streptomyces is the name of one, so you might see this on the table that we have in the study guide. Streptomyces produces many of our antibiotics, so they could be used in antibiotic production. Actinobacteria are noteworthy also for producing sort of filamentous colonies. And that's where the name kind of originates from, is that they're sort of fungal-like in that. Many of these are also soil bacteria. We find a lot of them in the soil, and so we find a whole diversity of them in a lot of places, but a lot of soil bacteria belong to this group. So that's the gram-positive bacteria, and we kind of started that a little bit, and we didn't really get in at all into gram-negative, so now this is what we're going to focus on. Gram-negative bacteria are almost strictly broken down by that small subunit ribosomal RNA. So we're specifically kind of focusing on this. And they're broken into two groups based on that, the proteobacteria and the non-proteobacteria. So the proteobacteria are all these related groups, and essentially the non-proteo are sort of everything else, the things that don't fit in over here. They're not necessarily related to each other actually at all. It's just that they don't belong over here, so they're just kind of a grab bag of organisms. And so we'll go through a couple of these and highlight some unique characteristics that each of them have. So first, we're going to focus on proteobacteria. Many of the groups don't have a nice, unique characteristic to say they all have this trait or they all have this characteristic, because it's based on gene sequencing. So we don't have always those things. But for a few of them, we do. For example, between these two, alpha and beta proteobacteria, in general, right, most of the alpha proteobacteria are oligotrophic. And what that means is that they do not require many nutrients. In fact, they thrive in environments with extremely low nutrients. So an environment that has very little nutrients, they do really well in. An environment that has a lot of nutrients available, they actually don't. All right, so alpha proteobacteria do that. We find a number of marine bacteria that are alpha proteobacteria. There's low levels of nutrients in the oceans, actually. So that you'll see that. The beta proteobacteria, then, in contrast, they require high nutrients. So sometimes referred to by this term copiotrophic. So they require copious or extreme amounts of nutrients in order to survive. A number of pathogens belong to this group as well. So again, these are all gram-negative bacteria. Some may be cocci. Actually, some of these are. Most of them, though, are rods. The overwhelming majority of this group are rod-shaped bacteria, the bacilli. But we do see cocci as well. So we have those alpha and beta proteobacteria. We could use nutrient requirements, which is something we're going to get into more so with metabolism and talking specifically about the terminology related to nutrient use. And it does tie into this, so this is where some of these names of groups will also come back later on. Now, gamma proteobacteria, I left a bigger space here because there's a little bit more to talk about with them. Now, a lot of the bacteria that are associated with your digestive tract, with your gut or intestinal bacteria, belong to this group. And so it's a pretty big group. It's a very diverse group as well. So while there are some pathogens in this group, there are also a lot of important bacteria that are part of your microbiome that also belong to this group, and they're particularly these intestinal bacteria. So just keep that in mind. A number of the gram-negative bacteria that we work with in lab, so a lot of our lab species of bacteria, and as we go through the semester working with different gram-negative bacteria, I'm not sure if all of them, but I'm pretty sure almost all of them all belong to this one particular group. So they're almost all going to be gamma proteobacteria, which are the one type of gram-negative bacteria, and so they belong to there. And I said there's many members of this group. Now, these last two actually have very few members. So the delta and epsilon bacteria are not really as diverse. So they're both small. The delta proteobacteria are noteworthy, or something noteworthy about them is that they contain bacteria that are sulfur reducers. sulfur reducers. That means that at the end of the electron transport chain, which hopefully you may remember, that oxygen is the final electron acceptor. So the reason you need oxygen is because it picks up electrons in your mitochondria at the end of the electron transport chain, and that is what actually drives the flow of electrons through the electron transport chain. That flow of electrons is what powers the active transport pumps that create a concentration gradient, and that concentration gradient through diffusion is what produces majority of your ATP. So ATP production in a cell is tied to these oxidation events, right? And in bacteria as well, while they don't have mitochondria, they're going to have an electron transport chain that we'll get into later with metabolism that is the same. The mitochondria evolved from bacteria. Now, sulfur reducers can actually use sulfur instead of oxygen at the end of an electron transport chain. That's kind of where the name comes from. So that's kind of a unique thing. So they can live in environments without oxygen or use sulfur and still run those same processes. So that's significant in that way. But there's not a lot of members of that group. And then the epsilon proteobacteria is one of the smallest groups with the least number of members. One of the things that they are known for also is another unique kind of metabolism. They are chemolithotrophs. Right, and so a chemolithotrope uses carbon dioxide. Again, these are terms that we're going to get into more so later when we go do metabolism, but it kind of feeds in a little bit now, so you get some tie-in with that. Carbon dioxide is the carbon source. And for electrons, instead of oxidizing organic compounds like sugars, which is what we typically do, they oxidize inorganic compounds. Organic oxidation. So epsilon proteobacteria have a little bit different metabolism. Again, all these groups can have a variety of different members that use or don't use oxygen. They could be different shapes and do all these different things. But and all members, one of the hardest things about this is all members of the group aren't going to necessarily display these characteristics. It's just that they're noteworthy. There might be some very important members of the group that do, and many of them may carry these traits. Right? So that's gram-negative bacteria. The biggest, largest group of gram-negative bacteria are called proteobacteria, and they're broken down into these subgroups: alpha, beta, gamma, delta, epsilon. So just kind of be familiar with those, something that you could just identify one or two little key things about each of them that might be unique about the group or make it kind of unique or different. Now we go into overall the non-proteobacteria that I said they're here because they're just genetically different and unique. This one group here, the spirochetes, is particularly obvious, all right, in a way. These are the spiral elongated shaped cells that have a periplasmic flagella. So they look very different than the others. They have this unique structure that the others don't have. They tend to be very difficult to culture and very difficult to treat with antibiotics. So there are a number of members of this group that are a little more dangerous pathogens that belong to the spirochetes. And so a couple of them are listed. Lyme disease bacteria belongs to this group. Some STD bacteria belong to this group. So they're noteworthy for that reason. CFB is actually 3 different groups of bacteria that are all kind of lumped together. And the C and the F and the B is the first letter of each of these groups. So the first group is the Cytophaga. The second group, the F, is the Fusobacteria. And the B is the Bacteroides. Now, of these, this one is the one I'm going to focus on mostly. They're going to come up now again because microbiome is going to be the next topic after we follow this up. This group here is going to make up typically about 30% also of your gut bacteria. So intestinal bacteria. So remember I said there's some gamma proteobacteria that are in your gut, and they're very important members, but a huge, huge amount of your gut bacteria belong to this group. And one of the things that happens to people who have imbalances in their microbiome is that the percentage of Bacteroides is altered in a way that is detrimental to those individuals. So it's really noteworthy for that. The cytophaga and the fusobacteria are often aquatic bacteria. Well, actually, these are aquatic ones. These fusobacteria can be found in your mouth. Typically, they're also both anaerobic. So these are groups that typically don't like oxygen. Right? Not really all these Bacteroides don't like oxygen either. So that's one kind of thing about this group, the CFB group as a whole, that you could kind of hold on to, is that none of these really use oxygen. They're mostly anaerobic bacteria. Water in your mouth, we tend to find this group, and then in your digestive tract. And the last group, this group here, the planktomyces, they are kind of odd bacteria in a number of different ways. The planktomyces, they have a holdfast structure, so they actually have something that looks almost like a plant root. Okay, so algae have a structure called a holdfast, which isn't a root but often looks like one, especially because they look plant-like. But it really just holds them, it anchors them down to a surface. So these are another group of aquatic bacteria. They also reproduce instead of by binary fission, which splits the cells in two equal halves. They can reproduce by budding. So actually a little bud will form a new cell. So the new cell that forms in cell division is actually a smaller cell. It's not the same size as the original cell. So planktomycetes are, so some of these names of these groups are just like a little blurb. It's just like the name of the group, kind of where it belongs, and a little example. And that's really all you need to know. You're not going to have to dive into it. As we go through the semester, and we talk about different things like the microbiome, like pathogens and so forth, we'll keep bringing up the names of groups of organisms that are important, say, on your skin, are important in your digestive tract, or are important pathogens. And then these names will come up, and these names will come up again and again and again. So this is why at this point in time, we're not getting into a lot of detail with each of them, but it's important that you just have some level of familiarity with the names of the groups, some general characteristic of the group, and kind of where they belong. You know, are they closely related to each other? Are they further apart? And that's pretty much it. So as far as studying, there is also a big long table with all these organisms in it, but there are some noteworthy ones. I would say you're just kind of looking for maybe one organism, if there are some names in it that belong to a group, or a characteristic or trait like this that belong to the group. Like if you can identify alpha proteobacteria as being oligotrophic, that's good. That's what you need to do. So that's all we're going to do with the classification of the bacteria, and I still have to do a little bit of a review for the exam, which kind of put some of the material together, but that will come separately. So if you kind of put this together, look at what is in really the book for this class, is one of the better ways actually of going through this material. So if you look through it, what it will do though is then give you just a real quick little short, a sentence or two, a characteristic of the name, and then it just mostly goes into names of species that belong to that group that are interesting or have something about them. So I wouldn't worry about focusing on the names of all the species that belong to each of the group, more just a characteristic or trait. Sometimes they don't even give a characteristic or trait. Sometimes it's just the name of a species. So in those cases, that's where you might want to say, oh yeah, okay, Heliobacter pylori is kind of mentioned with this particular group, so I'm going to kind of remember that name or associate that name with the group. Okay? So that's all we're going to have for the taxonomy of bacteria for now.